Okay, there we go. Okay, so hello everybody, welcome to the next Mapping Ancient Africa seminar, our fifth in this online series, and we're going to be focusing on past vegetation, the African Pollen Database today, and we have, Rahab and Sarah and Lynne have been putting together our presentation for today. I think Lynne is about to share the screen and talk to us. Is that correct?
Sarah is going to
talk to us first.
So this is going to be kind of like a three-part talk where I'm going to start and I'm going to talk for about 20 minutes about the African Pollen Database. This talk was sort of predicated on the idea that some of the ideas that came out of our very first workshop for this project, where we have experts from a variety of different fields. We want to think about developing questions and ways of putting all of these different data streams together. But I know that I felt and other people felt within these communities that it would be useful to get a sort of sense of like, what are the different data types? What do they look like? Like, what are some of their quirky attributes that you have to work around? And like, how available are they? And so this is kind of the paleoecology, African paleoecology side of the project's version of that talk. I'm going to start and talk a little bit about our efforts to relaunch the African Pollen Database and some kind of data quirkiness involved in those data. And then Lynne and Rob are going to talk about some recent applications and advances in African paleoecology that I thought were really nice examples that will, give some ideas, I think, as well. So I'm going to share my screen here. That's not what we want. We want this. All right, so bear with me while I get this organized. Okay, so that was the motivation for the talk. The purpose of the talk is just to give people kind of a sense of what data is available. And for people that aren't paleoecologists, but also for the paleoecologists as well, a little bit of a primer on the data structure of the African Pollen Database, some of the challenges, and I'm thinking of these challenges within the perspective of like both challenges as well as opportunities, and then some potential applications from Rob and Lynne. Okay, so a kind of general overarching sense of why should we care about having data in a database, why put your data into a database. Sort of threefold reasons, for most germane to this project would be the first thing that I have listed on here, which is that, we want to come together and answer some science questions. And those could be questions that aggregate data from just a few sites or a single site and think about how species are responding to climate change. But ideally, from a paleoecological perspective, we would want to increase our, network of spatial resolution and be able to use paleoecological data sort of within a big data framework. And that really requires us putting data together in a database. The second reason, and I think this one is really important, is reducing data entropy. I have a figure up here that basically shows time on the x-axis and why information content of a particular data set and metadata along the y-axis. And you kind of can get a sense that never is a data set more information rich than at the time of publication, but that over time details are lost by the individual PIs that generated that data set. There might be an accident that destroys data completely. Someone retires, and sort of slowly these details begin to be lost and are completely lost oftentimes with the death of the investigator. So, putting data into databases when at the time of publication, when we have the most access to information, is really important for, continuing that information on into the future. And the third thing that I'm going to talk a little bit about later is, and that is really important when you're trying to synthesize data and ask big questions, is having data that's standardized and also accessible. And so I have this acronym FAIR up here, which refers to best practices in data standards that were developed in a paper by Wilkinson et al. In 2016. And now you see kind of all throughout the geology world in terms of a model for best practices for how to make data accessible to everyone. And that acronym is that we are meant to have data be findable, accessible, interoperable, and reusable. And all of these things put together makes me feel like, putting data into databases, developing databases, is best for individual scientists. It's best for our community of scientists, and also, for doing good science that has like important practical implications. Okay, so just a little bit about the history of the African Pollen Database. It was first developed in '96 when there was the very first meeting of the community that put the database together, that sort of developed a data model for principles of the functioning of the African Pollen Database. And then from a period between about 1996 and 2007, this was an effort that was funded by multiple different international organizations that then did a series of different things that put together the different parts of the African Pollen Database. And so this would be what you might think of most commonly when you think of a pollen database, and that's collating all of the modern and the quaternary pollen data. But there were a lot of behind the scenes important components to this as well that were important for standardization, like standardization of the pollen nomenclature, which I'll talk about later, the creation of new age models, training of scientists, and also the development of a web tool, a really important web tool for the identification and digitization of pollen images. And then they also developed a lot of links to global pollen databases as well. So unfortunately in 2007, the outside funding was withdrawn, and the French data center that was developing the APD as a website closed. And so APD activities lapsed in 2007. At that point, new data weren't able to be integrated, and the website became obsolete. And since then has been very difficult to access and aggregate African pollen. So that brings us into the Relaunch, which was funded through an international organization, the Belmont Forum, through a call for science-driven e-infrastructure innovation, and a project that was funded that has been entitled Abrupt Change in Climate and Ecosystems, Where are the Tipping Points? And so that project began in 2019. It's continuing through 2022 and is likely to be extended for a couple of years. The sort of general goal of our side of this project that touches on this project is the relaunch of the African Pollen Database site itself, as well as the integration of the APD into Neotoma as a constituent database. But outside of like the actual like infrastructure development part, one really important part of this project was actually reinvigorating the African paleoecology community, of which this Mapping Ancient Africa project is sort of a direct line from that, and do some really cool science as a part of that. So some early activities in getting this effort up and running again from 2019 to 2020, when the pandemic started, were a couple of workshops, both in France and in the Netherlands, that reconnected African paleoecologists from all over the world, and helped us sort of discuss the state of the science, rethink protocols of the database, and also some best practices for using the data. There's been a lot of data, new data that's been published since 2007. And so there's been sort of ongoing efforts to acquire and collate and validate new data sets, as well as the old African Pollen Database holdings, to update the APD nomenclature, which is the sort of standard naming of pollen taxa for at a continent-wide scale. And then there was a big special issue that was recently published, of which Will was one of the editors, in Paleoecology of Africa, that would be a good reference for anyone interested in this data, because there are sort of a few papers on broad scale what the state of the database are, as well as more regionally specific research papers that present some really good examples of applications of these data. So the sort of vision of this project would be that at the relaunch of the African Pollen Database, we would develop a data model that thinks of the African Pollen Database not just as a website, but as a community, as well as a constituent database within Neotoma. And so I have the sort of APD data flows on this figure right here, this flow chart that sort of shows the APD data, starting with a committee of paleoecologists that are data experts that are validating that data, converting it into 2 different formats that get uploaded onto 2 separate databases, the Neotoma database, APD holdings within the Neotoma database, and then an African Pollen Database standalone site. And that the holdings between these 2 different databases are actually identical and they're interoperable. They exchange back and forth using a lot of the services within Neotoma that we're able to leverage through this relationship. And that then these data are available through these different formats to be used within R and other statistical packages by this community of data users and members. And so the goal is, like I said, to create these 2 separate databases, but that are interoperable, that have high quality curated data within them in order to support the African paleo community within a model that's hopefully more sustainable for the long term, so that we don't have a lapse like we did with the previous version of the African Pollen Database. And also develop a governance model that encourages participation by new members, contributors, and also research communities, of which the Mapping Ancient Africa project is a great example of one of those. So in brief, we've formed a governance council that's setting policies for the APD data model, with a meeting coming this summer to vote on those things, developing bylaws and data usage policies, which I'll talk a little bit about later, and then agreements with other databases as well. And then training data stewards. What's a data steward? A data steward is someone that helps get data contributions from scientists that are generating data, as well as doing a lot of quality control processes and age model development processes in order to really make the data that go into these databases like as standardized and high quality as possible. Okay, so what's currently going on? We're sort of in a pretty advanced development stage for both of these different components of the African Pollen Database. There is an African Pollen Database standalone site that was developed through Anne-Marie Lazine and an engineer that's working with her. The link to that database is posted there if you want to go take a look at it. It includes a few different parts: the pollen data itself, modern pollen data, and fossil pollen data, with a total of 397 data sets that have chronological information, then additional 83 that don't have any chronological information. And then that pollen identification tool that I talked about is also on that site. And then we're in the process of then integrating those data holdings into Neotoma. The data stewards that I talked about before are mainly who's involved in that process. That is ongoing. We currently have very little modern pollen data on Neotoma, but quite a few fossil pollen sites, 223, 50 more pending, so almost 300 sites, fossil pollen data sites on Neotoma that are like readily available in Tilia format if you're a Tilia user, in CSV if you're not, and that is also integrated within the R environment through a package called R Neotoma, and so you can use and manipulate and visualize data in R as well. And then I also wanted to mention we've also identified 247 published records that are not included within the APD database right now. So I think, we're hoping that we're reaching out to people and hoping that those data ultimately are contributed. I just wanted to give people a sense of how data rich some of these data sets are. And so this is a little screen film that I took of Tilia, which is what we use in order to validate and upload data into Neotoma. And you can see I download a data set, a single site from Neotoma, and then you can see all of the actual data in raw counts are downloaded into that environment. And then I sort of go and scroll through all of the metadata that's available as a part of those data sets as well, which will come back up here in a second. So this is the raw data as well as charcoal data that we have associated with a lot of sites. And then, these records are very metadata rich, which is really the point that I wanted to make here. Many new records with really good chronologies and age models as well. And then on Neotoma, if you are using Neotoma Explorer, you can look at individual records on Neotoma Explorer, and they automatically give you a sense of what those pollen data look like, because it will calculate abundances, like a more sort of small pollen diagram for you automatically. So that's what those data look like on Neotoma, as well as through Tilia. As I said, they're also available through the R Neotoma package. I also wanted to think a little bit about some best practices for paleoecological synthesis, since that's what really touches on the development of what comes out of the Mapping Ancient Africa project. And I wanted to think a little bit about, what some of the constraints that I felt, based on the data there were for developing questions, especially interdisciplinary questions. And within that, thinking about like leveraging the strengths of the data, especially the new data, while being aware of and addressing some of the challenges that I see. So there are a lot of strengths, actually, like as I've alluded to, based on the numbers that I told you of fossil pollen data sites, both in the APD standalone site as well as available on Neotoma. There's a huge amount of data available that follows these FAIR data standards. So the data is standardized, it's been validated, it's been cleaned, and it's like open and available. So it's easy to find, it's easy to visualize. And you can use it interoperably with a lot of new tools like Lipid Playground, Neotoma Explorer, are all sorts of different platforms. And so this just gives you a sense here of this is what the holdings within Neotoma looked like in January 2020, before a big effort for data mobilization. And then I took this screen capture on April 5th. And you can see just how many new points are up on that map now. There have been a lot of methods advances, like data in terms of geochronological dating. There are a lot of new records that have very high precision chronologies, as well as very high sampling resolutions. This is a great example of a record published by Graciela Gil Rivera from Ethiopia. Will, can you mute someone? Thank you. Okay. So this is an example of a new pollen record that's now a part of the African Pollen Database. And this paper was published as a part of our special issue from Ethiopia. And you can see it's a record that covers about the last 14,000 years. Extremely high temporal resolution in the pollen sampling, as well as in the chronology. It's also one of a new generation of records that includes a lot more than just pollen data. So there's a lot of non-pollen palynomorphs included in these new pollen data sets, charcoal information. A lot of sites not just have pollen data, but also have paleoclimate information and sedimentological data as well. So there was a lot of prior work with the previous APD holdings prior to 2007. And I'm not going to go through this list of, all of these different sort of like types of research questions, paleoecological research questions that were evaluated with previous APD holdings. But, these areas are really ripe for reassessing with this new, more metadata and dimensional paleoecological data, in addition to a lot of new questions as well. Some challenges, just really quickly, there's a variety of scientific questions that you could skim the literature and add a lot more things to this list. There are still a lot of aspects of ecosystems that are difficult to reconstruct with pollen data. We do composition and structure really well. Diversity is more of a challenge. And there's of course issues with constraining things like dispersal and pollen production, as well as variability in the representation of vegetation in different biomes, especially in places where there's a lot of insect pollinated plants like the tropical forest. So there's a lot of things that we want to look at that are maybe still invisible. So we have to leverage the things that we can do. There's a lot of, as I alluded to, like sort of quirky data and metadata attributes that I think people should be aware of. We have a lot of new pollen data sets, and we have a lot of old pollen data sets as well that can be really useful, but that also, have some other constraints. So our oldest pollen data set was produced in 1954. Many of the older data sets have no chronology or like maybe one conventional age. So being aware of the time that these data were produced is really important. Spatial and temporal scales really vary. You can see from this map of our APD holdings that we have a lot of records from South Africa, a lot of records from East Africa, and sort of a smattering of things in like West and North Africa. But it's not a perfectly continuous sampling in space, and neither in time either. And so this plot just gives you sort of a general sense of what is the frequency of APD records by their maximum age. And so this is out to 1 million years and out to 100,000 years on the bottom. Most records are younger than LGM in age, and many, relatively low resolution as well. So it's just something to take into account. They're multivariate assemblage data, which means that, they are by nature very sparse data. They're non-normal in their distributions. And so that needs to be taken into account when you're doing any statistical tests. They're also unevenly spaced in time for the most part. So there's probably more quirky data attributes that really, constrain the statistics that you can do with some of these things, but that's just, kind of the high level stuff. There are issues in standardization as well. We're doing a lot of data cleaning, but databases are full of errors. And so careful data filtering needs to be done prior to any meta-analysis. New age models are being made, but there's still a lot of older age models associated with these data. So it's important that people look at them from an individual record perspective. And then I'm not going to go into detail about taxon names, but taxon naming in palynology can be really hard and vary between labs and also vary between region. So there's been a big effort to try to standardize and harmonize taxon names at the continental scale. And one great example is a pollen type called Isoberlinia pollen type. You can see all three of these different pictures of pollen grains fall within this type, but they are all from actually not even just different species within that genus, but from different genera. And there are actually 15 different species in 9 different genera that fall within that pollen morphotype. And the reason that it's important to harmonize or standardize pollen taxon names is that within many regions today, in the example of Isoberlinia, you can find genera of plants that co-occur with Isoberlinia, but that don't actually grow within the same temperature space. And so this is just a couple of figures from a 2007 paper that did this assessment with mean annual temperature, mean annual precipitation along the y-axis, mean annual temperature along the x-axis. You can see that these dots are different species of Isoberlinia. They have a tendency to grow in more seasonal, low precipitation climates. The other genera within that morphotype have a tendency to grow in wetter climates. So neglecting that distribution and not standardizing pollen names can lead to non-negligible errors in continental level reconstruction. And so there is this APD standardized taxonomy that was developed first in a paper by Vincent et al. In 2007. It was updated in 2021. And there was a recent release of a new fully up-to-date APD taxonomy that includes information about the biology and the ecology and the phytogeography of all pollen taxa within the African Pollen Database holdings. So if you're interested in that, let me know. I'm not going to go into a lot of information or detail about the new opportunities for using these data, because I think Lynne and Rob are going to talk about some really good examples. But there are a lot of new tools for processing and standardizing pollen data, including the R-Neotoma 2.0 package, which was just released. There are a lot of packages that allow you to work with data, Neotoma data directly within R, like Bacon for age modeling, Harmonizer for harmonizing taxon names. And there are also a lot of new opportunities based on analytical tools for working with Neotoma data or lipid data in terms of new and kind of standard R packages that work really well with really standardized paleoecological data. So that's not an exhaustive list, but it does give you a sense of some of the opportunities that are out there. And then there's a lot of overlap with other efforts too that are currently going on that I see as being sort of like really interesting potential opportunities. There are a lot of sites, like I said, that have independent paleoclimate data sets that really could be useful to leverage, and as well as new data syntheses of temperature and precipitation at a global or continental level. There's a lot of overlap with some of the efforts of PAGES working groups, PMIP and CMIP efforts, as well as mobilization of other data types within databases like Neotoma, as well as other developing databases like the PaleoFire database and the Paleobiology database. One last challenge that we've identified is buy-in. We have quite a few data sets that have been published that haven't been contributed yet. So the last thing that I really just want to say kind of quickly is that there are a lot of advantages, not just to the community or data users for contributing data, but to individual scientists as well, such as visibility. A contributed data set gets a DOI, a direct object identifier, which is like a website, essentially a website for that data, which makes that data much more visible. Your sites get included in big regional syntheses, and that can ultimately be really useful for you for understanding the larger context of your own site. It looks good to funders and publishers. Your taxa get integrated into the APD taxonomy, and then your data is safe. You don't have to worry about version control or like data loss anymore. It's safe. But attribution and credit are really important, I think, for some of these issues of buy-in and data contribution. And I'm not going to go through these data usage policies, but I did want to just mention them because I do think they're important for a big project like this that might be leveraging some of these larger holdings within these databases. And that's, regarding citation, there are data usage policies both on the Neotoma website and the African Pollen Database website about citation of databases, PIs, as well as policies for co-authorship and what like a rule of thumb for co-authorship. So if you're using the data, I just encourage you to look at those as well, because they're really important. So in summary to what I wanted to talk about, APD data are increasingly available. They're really easy to use, both on Neotoma, on the APD website, within R. They're very standardized. They're basically like ready to go at this point. But the usage of them should come with the awareness that they have some strengths, as well as some limitations that I've already talked about. And that the best practices for using them involve standardization, quality control procedures, and awareness of those data usage policies. But there are a lot of new tools that are emerging that can help with those things, as well as developing new questions that like really weren't even answerable, or that weren't answerable with the data that was available prior to 2007. So there's a lot of opportunity for reassessing questions that have been asked in the past, as well as for asking completely new questions. So with that, I will stop sharing my screen and I will pass to Rob then, I think is going to go next.
Thank you, Sarah.
Thank you, Sarah, for that. I'll try to share my screen.
Yeah, we can see that nicely.
Okay. Yeah, so following on Sarah's presentation, which she really emphasized on the applications and the opportunities that we have with the APD and Neotoma database, I'm going to narrow down into one of the projects we are working here on the application of the paleoecological methods to meet the social scientists, so that we can be able to see if we'll be able to have and guide on some policy making, especially on the things that are happening between human and environmental conflicts. But before I get there, I want to emphasize on the opportunities that we have in East Africa and what has East Africa contributed to the APD database. Realize we have a total of 250 sites that are representing different depositional regimes, lake sediments, some of them from swamps and peat, archaeological and marine. And we have half of those, they have chronology, and half of them, they do not have chronology. So as Sarah had pointed out about the opportunity of trying to figure out what is happening in the region with these data sets at the APD, and if we keep uploading more of what have been dated and what have been analyzed, it will give a bigger opportunity for people who are trying to see on regional scale what is happening across the space given any time. So like in Kenya, we have a total site, at APD, we do have 51 sites, and at least 59% of those sites have chronology. And then we have, it's not moving anymore, but what's happening?
Yeah, we can see a nice leaf, Rahab. Is that the right screen?
No. I think I've missed out. I should stop again and then share again.
Okay.
Can you now see the screen?
With
East Africa contribution to APD?
No, we just see it. We see a very nice fossil leaf.
That is my desktop. Let me see what I should do so that I can have. And now?
Still the same, I'm afraid.
Jeez, okay.
I can see your cursor moving around.
Let's see. Oh, okay. Something happened and I'm not able to get to the desktop. Sarah, you still have it? Oh, okay, let's see.
When you press share screen, you have an option to like either just share your full screen versus like an application. Are you picking just the screen itself, like the desktop?
Yeah, let me first stop that. Sorry about that. So should share. So far you haven't seen any of my slides?
No.
You see anything?
Is the PowerPoint open, Rahab?
Yes, it is open.
And if you go share and then you click on the PowerPoint and not your desktop?
On the PowerPoint.
Does that come up in when you click share? Instead of saying screen.
Okay, let me say share screen.
Right, no, still the back.
Is it there now? Participants can see? No, same thing.
Same thing.
So you sent me a previous version and I just downloaded it. Do you want me to share my screen?
Yeah, please, kindly.
Okay. I think it doesn't have the map in it. It's the one that you sent before.
You don't have the recent one.
I think I have it, Rahab. I can share it.
Okay.
Okay, please.
Let me just.
I figured out this thing will happen for some reason.
I'm getting there. Let's.
Okay, so if you stop sharing your screen, Rahab,
Yeah.
Okay.
We'll switch over to Lynne.
It's loading. Hang on a minute.
It gives me a chance to check out all the numbers of people here. So we've got 28 participants, and there's a whole range of people from all over the place. So it's really great. Great to see a nice turnout again.
Okay.
Now we see it.
Okay. Thank you, Lynne.
Just tell me next and I'll move on.
Yeah. So as I was explaining and following up on Sarah's presentation and the opportunities that APD Neotoma database present for us paleoecologists and all those who are studying how the vegetation environment have changed over time. So I'm going to present an example of a project that we are undertaking here in Kenya for trying to meet the paleoecology and social scientists to see if we could be able to address some of the current human environmental conflicts. Next. Next.
Sorry, I'm hitting next and nothing's happening. Come on. There we go.
Yeah, so hit like 3 times, yeah. So to bring especially the East African into context to see what we have in East Africa represented at APD, we have a total of 250 sites that are representing different depositional regimes. We have lake sediments, swamps and peat, and archaeological and marine. And half of those sites have a chronology, but half of them they do not have. That is from the East African in general. And it's a call that all those people who have those data sets, they can try our best. Then we upload these data sets to the APD because it presents a good opportunity where we can be able to compare what has been happening on regional scale in these countries. In Kenya, we do have 51 sites, and half of them are already with the chronology. In Uganda, we have 43 sites, while in Ethiopia, we have 39 sites. So if you look, you will see most of them, we are ranging between 30 and 40% and 50% for those sites that have chronology. Next, Lynne. Next. Yep. In Tanzania, that is the more south side, we have 49 sites and 23 of them have chronology. While Burundi and Rwanda, we have, I think this is where we have few sites that have been analyzed and have been uploaded into the APD. Next. We have also Congo and we have Malawi and other sites like Indian Ocean. These are marine corals and Arabian Sea. All these have been uploaded. And the big message on these sites is that these data sets are available. And we have one example that is work in progress with Rob Machat and Esther Gdombi and myself. And also we have Collins and others. We are trying to look at how the mountain have changed over time, the East African mountain, and looking at the biomes, how they are shifting along altitude over time. And mostly we are looking at the glacial time. Next. So coming to the main highlight of today is to apply the paleo data to be able to inform the current environmental human conflicts. Next. Okay, so these are just images that I had brought to show what the current conflicts that are happening in Kenya in Mau Forest, where people have been living in the forest, and there's a lot of degradation. There's a lot of negative impact on the drainage systems in the Rift Valley region. And it reached a point where the government realized that they're going to lose a lot of biodiversity by allowing people to continue living in the forest. But because many people, they want to hear exactly, they feel like they are not impacting negatively on these ecosystems. So we thought if people can think of how they can bring hard data or scientific facts to be able to explain to people how bad the situation is getting and how bad situation will even get worse in the future. So we, looking at those different scenarios, we wrote a proposal to British Ecological Society, which was funded, to look at the recent time of degradation, how humans have changed the ecosystem, and especially the forest ecosystems, which are very sensitive to occupation, to human occupation, to human grazing, and all the things that happens, activities that are going on in the forest. Next. So next again, so that I show the site where we are currently working in Aberdare. Forest is one of the forests that was affected immensely during the colonial time. And this forest has a long history of human occupation and movement in and out the forest, and different governing structures have impacted the forest in different ways. Because before colonial time, people used to go for foraging and hunting. They never used to do a lot of cropping in the forest. But when colonial government came in, they pushed people now back to the forest, because this is in one of the very fertile areas we have in Kenya. So these lands were taken for tea plantations and coffee plantations. And then the local people were pushed in the forest, where they were doing now their cropping and grazing within the forest, and as well staying in the forest, or occupying the forest. But now after the colonial time, after independence, then they were evicted again from the forest by the government that took over, so that they can be able to restore the forest and to avoid a lot of human and animal conflicts. So, and these forests have all these sites that you walk through, and you can see literally how the vegetation was impacted, how grasslands were created. Those spots, grasslands within the forest, were created by this occupation. Next. So, and we are analyzing pollen and phytolith at different localities to see if there will be any signal of change in climate change in temperature, by seeing how the poedic grasses, C3 grasses will change, and how the C4 grasses are changing. And then also pollen will give us a bigger picture of what is happening with different species and along the altitude, and even whether we'll be able to get the crop pollen for crops within these sites. And then we have archaeological and historical data that we are also collecting. We're collaborating with archaeologists like Dr. Diema and his team, because they are able to identify even how the fencing and how the people were able to demarcate these forests as they map them for themselves, as they owned different areas for themselves. Next. So some of the finds that we found that were evidence of historical occupation and archaeological aspect are the things like the keys. These were things that were never there before before the colonial time. So this, if you see some of these and the coins that we found, they could tell us that truly these people were living there. And then we raised short course 50 centimeters using the Livingstone coral, and you could see how the sediments have changed, or the swamps have changed, to some of them drying completely at some point, but now some of them are kind of restoring back and getting back to their former situation. Next. And the other thing about this project I need to point out is the involvement of the students, because this is a field especially in Kenya, that attracts very few students because of I think the nature of the work, most of the time in the lab and all that. But involving them in the field, they have come to appreciate the techniques. They have come to realize what other avenues can be used to also do science, and again be able to inform the community, to be able to inform the government of the dynamics that are happening and what is the way forward. Next. For the now so far, we raised around like 20 short calls, and from them we sampled 800 samples, which were subsampled at 0.5 centimeters. And from each level, we analyzing phytoliths and pollen and spores, because we want to see the impact of herbivore, including the animals in these sites, so that we can be able to comprehensively be able to inform. And so far the work is exciting, I must mention here, because we have things like cereals, maize, and sorghum that are happening in the times that which is making a lot of sense. Next. Yeah, so these are the key indicators that we are looking at on those images. We have phytoliths from sedges, we have phytoliths from seed grasses, and we have others from the woody components. Then from pollen, we have Biden and Asteraceae that are the key indicators for the changes in these environments. Next. Yeah, so again, it has presented a training opportunity for undergraduate students. It's not only in the field, but also in the lab, teaching them a few techniques on how we process and how we get to extract phytoliths, and how we get to extract pollen, and even identification of those proxies. Next. Yeah, so I want to take this opportunity to thank all the team players that are in this image that participated in the project, and they've made it to be so far a success. And we are looking forward, in a few months' time, we'll be having a few calls that are already complete with analysis that will be presenting and to the people. Thank you. I guess that's my last slide.
Rahab, that was great. Lynne, are we switching over to you now?
Yeah.
Okay, great.
Okay, can everyone see that? This is a very brief synopsis of the current directions of paleoecology research in the Cape Floristic regions. I'm just going to present today, and the work that we're doing at the Paleo Lab at Nelson Mandela University with all our collaborators across the world as well. Just to briefly go on about how important it is in terms of the area. The Fynbos biome, otherwise known or synonymously as the Cape Floristic region, is a biodiversity hotspot, as well as the two neighboring regions, the Succulent Karoo and Maputoland, Pondoland, Albany Thicket region, and we're trying to expand our research directions into those two neighboring biomes as well. It's also, so from the botanical perspective, it's really important. From a climatic perspective, it's important, and also really complicated and complex in terms of the long-term climate changes in the region due to the many interacting features of the climate system that sort of like interact within the region, and the fact that we have the winter rainfall zone, the CFR encompassing much of the winter rainfall zone, which is very different from the rest of Southern Africa, which is currently experiencing summer rainfall. We were quite lucky that we have not only just sediment cores extracted from wetlands within the coastal regions, but we also have higher expedient sites in predominantly the Cape Fold Belt Mountains, so the montane inland regions of the CFR. We're working on both those angles to extract paleoecological data, particularly pollen. In terms of the paleoclimates, we can definitely say that it was not straightforward and quite complicated, and ranging over different spatial and temporal scales. We have different manifestations of the different climate systems within the region. And what has come out of the recent, about the last 10 years, research by myself and many other peoples, including Brian Chase in terms of the Midden work, is that there's a mosaic of distinct climate subregions within the CFR. And really to tease out some of these nuances, we need new high resolution terrestrial and marine records. And this is where we are working on at the moment in terms of generating more records for the area and strategically looking at different types of proxy evidence from these and what we can pull out of them. And so this is just a map showing sort of the future potential and the current work that we're doing at the moment. In terms of the brown dots are the higher experiment sites from Buyin that we are processing at the Paleo Lab for pollen and other paleoecology proxies. And then the blue dots are the wetland cores that we have very recently, over the last year or two, extracted from potentially predominantly the Southern Cape Coast or Southwestern Cape Coast. And that's all happening at the moment in terms of generating data. We're also trying to bridge the gap between paleoecology and ecology in terms of looking at a more analytical ecological lens to the existing published CFR pollen records, as well as the ones we're currently producing at the moment. And two key initiatives sort of fall within this category. I'm working together with my students with the PI of a project that's looking at discriminating Asteraceae pollen and producing a Asteraceae pollen key for Southern Africa. And that's Dr. Saul Manzano, who's in Spain, but he's working with Sanbi here in South Africa. And this is my student Luke, who's measuring tiny little bits and pieces of daisy pollen there with our high-end research microscope, and also looking at it through the lens of SEM imagery. And this is really important because Asteraceae really dominate many of the pollen records for Southern Africa. And we really don't have, at such a large scale family level, we can't really pull much ecological or paleoclimate data from the indications of Asteraceae at the family level. So with this higher resolution, finely tuned key, we can re-examine some of the existing records, answer some more nuanced ecological questions once we have this key in place. But that's a little bit more work on that front, mainly so Will needs to do a lot of measurements himself. We're also doing new pollen vegetation calibration studies, which is something that hasn't really been done in CFR at all until now. And thanks to Adele Julia, who helped myself and a postdoc make a whole bunch of pollen traps and go out into the field last year and this year. And these are this is the distribution of the pollen traps that are out there, or the ones from the Southwestern Cape we've just retrieved a couple of weeks ago. And we had a couple of instances where baboons have stolen our traps. And so we've lost one or two of those. And then we put out some new traps in the reserve in Gqeberha, in Port Elizabeth, right next to my lab. We're quite lucky to have quite an extensive reserve there, and coastal thicket and fynbos vegetation that we put up in some key types there. So those we will retrieve next year, and a master's student of mine is working on this as her main project. And then we're also working on tying into the archaeological record and collaborating with some key archaeologists and new research projects that are happening within South Africa. We have a really rich archaeological history, and the key here is to sort of get a better idea of the environmental context of the archaeological assemblages and excavation research that's been done. And also in terms of sort of more of the recent history, the late Holocene, we're working on new paleo proxies such as coprophilous fungal spores and macro and micro sedimentary charcoal, combining this with pollen to look at whether we can see patterns of pastoralism and herbivory within the Cape records. And that is also being worked on by some of my students last year and this year. And those are the two main archaeological sites that I'm involved with at the moment, Buyin Pass and some Cape Coast interior, and a new collaboration with the PI, Alex Mackay from University of Wollongong in Australia, on the Farsh Rafia in the Suckling Karoo. So this is bringing into what I said in the beginning in terms of expanding into these other hotspots regions. And I just put in a grant for some wrecking for Hyrax middens and potentially some wetlands, although that's possibly not going to be very conducive because it's really dry up there. So to find any wetland deposits that will be useful to us might be a bit more of a challenge. And then all of this, of course, is feeding directly into the APD, and wherever I get a chance, I'm trying to upload all my data sets. I think I'm nearly done with that. And also prioritizing some key uploads for sites that, after doing some meta-analysis of the database that I have, that are key to put up there, are regionally representative and have good chronologies and those type of things. But really thanks to Sarah and her team for really leading the charge, and every week having meetings and encouraging us all to get connected to the APD through her training. And so that brings me on to really, if you have any Southern African pollen data that you'd like to contribute, please get hold of myself or Gemma Finch, who are the data stewards for the sub-region. And we really just need raw counts, but get hold of us, and whatever you have, we'd be very grateful to have that within the data set. And then again, like Rahab mentioned, student training and mentoring is a key focus, capacity building through my lab. And over the last 2 years, we've got several students involved in these initiatives and are working on key elements within these different projects that are ongoing at the moment. And I would like to thank them all, and some of them are here today on this meeting, in terms of their hard work and their just general enthusiasm for the subject. And that is it for me.
Thank you, Lynn. Thank you, all of you. Yeah, great to see the training, and great to see the dots on the map appearing. I think that's one of the really nice things about this initiative is just to seeing that African sites appearing on the map. So do we have any questions for Sarah, Rahab, or Lynn after that tour de force across the pollen of Africa? Either shout out or raise your electronic hand, or otherwise I'm going to do a slightly unorthodox thing, because I know Adele is there because she tweeted, she mentioned a message just now. And one of the things that was mentioned early on by Sarah was the modern pollen contributions into the APD. I don't know if Adele, I know you've written a very nice recent treatise on modern pollen studies across Africa, and just wondered if you wanted to comment on that, on sort of data availability and what was out there, and what the potential is of getting that transferred it up, just to put you in the spot, because I feel I can do that with you.
Thanks, Will. Yeah, so that is one thing that I sort of, when we were putting that big review together, I, in my mind, was like, these data should all be on Neosediment African Pollen Database, but yeah, ideally I'd like to do that. Now that I have a permanent job and my life is hopefully not completely on fire all the time, I might be able to get some of that in. A lot of it, though, were not published data sets, so it was very much we were collating what people had said about the data. It wasn't necessarily that the data themselves were available. So a lot of that was kind of based on, yeah, kind of obscure references to data sets that may or may not exist. So it's a sort of, I think, going through those references and kind of trying to pull out data sets that are complete enough to be put on would be good. Maybe I could get a master's student doing it at some point.
Yeah. Or just...
I can actually, I will have MRes students soon. I could maybe get one of them. Ooh.
Thanks, Rahab. Rahab's heading off to get her bus. Thanks, Rahab.
Bye, Rahab.
Bye. Bye.
Questions to Rahab via email? Or,
Yeah.
Or
Need to
load it up.
Okay,
Yeah.
bye. Thank you all. Thank you, everybody.
Thank you.
Bye.
Yeah, now you can motivate students, but also it's about, getting that net. You know who those people are who have those data. So then
Maybe
the people who wrote the papers that you got the data from. So
slightly, yeah. Hopefully some of it might be accessible, but a lot of it, I don't know, just wasn't there. We had to go on what was said about it rather than the actual data itself, unfortunately. I think Anna Marie Lazine sent me quite a bit of it, so I could maybe dig that out and see, but I'm not sure if that had already been put onto the database or like what stage it was in that kind of workflow. I wasn't sure where some of those data were, but maybe we can kind of regroup on the modern stuff. I don't know if Sarah knows more about where the modern data sets are.
So the modern data sets that were a part of the 2007 database, APD database, are now on the new APD site. I don't think any new modern data have been added to that. This is where things like making them identical becomes a little bit complicated, because there have been some new things I think that you have maybe even uploaded to Neotoma. So I think there's more new stuff on Neotoma, and then all of the sort of like 1,985 original modern pollen samples are on the old site. So at some point, basically, they need to be like merged and duplicated.
Interesting.
And I think I have a list of all of those, all of the data that we think should be modern data within the database. And so we could, I could send that to you, and you could see if that marries with what you think the case should be.
That would be useful, yeah, because maybe there'll be gaps or missing bits in either the review or the data. So yeah, that would be probably a good thing.
Yeah, that'd be great.
Yeah. And then if you send me that, and then yeah, maybe I can think about putting a master's proposal together to get a poor student to come in to come and do some analysis and kind of get everything uploaded.
Yeah, and if you, so I mean, I have some online training stuff that is available for students. And so since you're a data steward, you could have a student who could basically self-train, and then, you know, you would sort of be supervising that, but the training part is already mostly done and put together. So if that would be helpful, I could like give you access to all of that.
Amazing. I think I have it. I think I'm already on that Google Drive.
Google Drive. Yeah, it's all on Google Drive, but I've like put more stuff in there, and it's now like there's like a README, and it's in like a reasonable order. So
I think I've seen, yeah. It's really cool. Thank you.
Yeah, sure.
Cool.
Yeah.
Thank you, Adele, for putting you on the spot. Anybody else? Any other questions, comments, thoughts? George, I think you're trying to say something, but you seem to be muted.
There you go. Secondary sort of mic mute. So for generating new pollen data sets, is there a generic format by which, when we prepare them for archiving, would be helpful to integrate them faster into the APD? Is there such like a layout?
Yeah, there's not actually, I mean, there's no standardized layout, but it actually is relatively simple in that you can just send the raw data to any Neotoma data steward, but so you could send it to me, and then like a publication, like a PDF of a publication that would have site metadata in it. But as long as we have just like the raw counts, then we can go from there.
Awesome. Cheers.
Yeah.
Cool. Anything else? Although, George, I'm going to ask you a question now. What data are you generating? What exciting site have you got coming online soon?
Well, hopefully sort of by the end of the year, Central Congo Basin pollen data sets from two sites, one in the Republic of Congo, one in the Democratic Republic of Congo, peatland sites as well. So yeah,
nice.
Hopefully it'll come up.
Yeah, good. Any other questions, thoughts, comments? In which case, I will thank everybody again. Thank you all. And yeah, great to be on here. We have, in about approximately 1 month's time, we have archaeology as a theme topic, and we have our next seminar. I can't remember the exact date. Does anyone remember the date and the title? Lynn, you're usually organized with these things. Put you on the spot.
I actually don't know offhand, but
it's approximately 1 month.
Tyler should know. He's
Tyler.
Presenting.
Tyler, yeah.
I think ours is in June. Isn't there a May one? Or
yeah, it might be a jump till June. Actually, you might be.
Yeah, I think it's June.
yeah.
Okay, so we will see you
Somewhere.
all back here in June, and we will try and organize a physical type of meeting where we can hopefully get people together, either in the hubs and all together, sometime in this year, calendar year, is the plan, I think. But we need to talk about exactly when and where and how we do that. So good. I'm going to stop the recording. If I can do that. Can I remember how to do that? Yes.